the positive bleach sample that I used changes to dark blue.
Okay.
So there was a light blue color change in one of the cuttings comparing to a dark blue.
All right. And if we go back to our earlier discussion, if that fabric has been out in the elements where it's been subjected to snow and rain, would that certainly impact it?
Yes.
Okay. What did you note next?
So I then performed a infrared spectroscopy analysis on the sample. So I dried down the water and then ran the instrument.
And what did you note there?
I was unable to get a very strong match. These instruments' results give you a spectra or a series of peaks and valleys sort of thing. And we compare it to a variety of reference samples in libraries. I was unable to identify anything that matched strongly with the sample.
All right, continue.
That finished my analysis for that particular item.
All right. Now, when we go to the second cutting, walk us through that second cutting and what you noted.
Oh, I'm sorry. That was for all three of the cuttings.
Oh, that was for all three.
The only difference between any of them was with the color test. One of them turned light blue. The others had no change.
I understand now. Thank you so much. All right. So then, based on what you had noted, what did you report by way of findings?
I was unable to identify bleach.
All right. But certainly in terms of just the varying findings that, you were able to establish, were there findings that would be consistent with the use of bleach if that fabric hadn't been submitted to the outdoor elements? And maybe that's not an appropriate way to say that, and you can correct me if you want to.
So I'll explain it so my conclusion states my scientific conclusion of all of this data, which takes into account the different elements that could possibly be in bleach and would be consistent with the chemical or physical components of bleach, but it also takes into account what kind of environment it was in and other environmental effects and ruling out everything else to say that this was definitively from this particular compound. In this case, due to the limited amount of sample or the diluted sample potentially, and the fairly weak results for a lot of these samples, I can't say conclusively whether it was consistent with bleach or not, because there are other elements that could be explaining these factors.
Thank you. I appreciate that. Now, did you analyze the shorts for the presence of bleach?
I did not.
Or the bra?
I did not.
Explain why not.
So when we were coming up with the initial testing plan, we knew that these samples were going to be, they were, I guess, out in the environment for a very long time. We knew that it wasn't going to be a actual sample like liquid bleach. And with our current instrumental and scientific capabilities, we knew it was going to be difficult to obtain data that would be consistent with for an identification or comparison. So we developed a strategy in which we tried to find the most likely items, being one of the clothing items, the shirt, and then the bottle that would presume to have maybe held a compound. And with the results that we obtained, we did not think any further analysis would indicate any useful information.
Thank you. I appreciate that. So then let's go ahead and let's go down to the second submission. What was the strategy that was developed for the white plastic bottle and then the pieces of the broken white plastic bottle that came in?
So I analyzed the whole plastic bottle with the idea that it would contain the chemicals longer potentially than a broken bottle that may have spilled and be open to the elements more. So I figured the intact plastic bottle would be protected from the elements more so than the other items. So I chose to analyze that item in particular.
Can I ask a question? When that white plastic bottle came in, did it have a lid on it, or was there no lid associated with that container?
There was no lid that I found.
All right. And then my second question would be this. Was there any contents within the plastic bottle itself, or was it empty?
There was what appeared to be soil and debris on the
Okay.
inside.
All right. So walk us through that same discussion.
So the same strategy applied to this bottle in terms of the scientific steps that I took. The only difference being, instead of taking a cutting and an extraction, I rinsed the inside of the bottle with water and then continued on just like I did the previous item.
Okay. After you had rinsed the inside of the bottle and you were able to collect liquid from it, then did you analyze the water itself to determine whether or not there were any elements within it?
Yes.
Okay. What findings did you make?
I apologize. The x-ray was not done on the water extract of the bottle.
Okay. All right. Gotcha. All right.
I
So
can
what
understand
various
that
tests
either.
did you perform?
He's making mistakes.
So outside of the x-ray, I did the pH test, followed by the color test, and then the infrared spectroscopy.
Okay. What did you know during the pH test?
It was around a pH of 7.
All right. Color?
There was a light blue color and some fizzling.
Okay. And then what do you mean by fizzling?
It produced bubbles.
Sure. Does bleach produce bubbles?
It can. Yes.
Okay. All right. And then finally, with respect to the infrared spectroscopy. Boy, spell that for our record.
Okay. INFRAED SPECTRoscopy.
There we go. All right. And what were your findings there?
Again, I was unable to find a strong match for any of our reference libraries.
Did that conclude your involvement in this matter?
Yes.
Your Honor, I have no further questions.
Mr. Waters.
Mr. Stepanski, I'll just cut right to the chase. Every item that you looked at or tested, there was no bleach found on any of it, right?
Correct. I was unable to come to a conclusion consistent with bleach.
Okay, so no compound consistent with bleach was found on any one of those items.
Overall, yes.
I mean, your result says bleach could not be identified on the shirt or white plastic bottle, right?
Correct.
Okay. As part of your profession or your job, are you asked to try to identify bleach often?
Not often.
Perhaps other compounds? Cleaning supplies, are you ever asked to do this?
Yes.
You've done it other than just this case, right?
Yes.
Okay, and the first time or the only time you were requested to test anything in this case was March 25th and 26th of 2021. It looks like you didn't author your report until August 30th of 2021. Does that all sound correct?
Yes.
Okay. And at no point prior to March of 2021 were you asked to test any other item in this case, right?
Correct.
Okay, so those 5 items that we're looking at in this report are the only thing you were ever asked to test.
Correct.
Okay, and who requested these items? Is it just this Mark Bethel with the medical examiner?
It appears there's 2 different submissions. So Mark Bethel would be the first submission of the 3 items, and then Jennifer Pullen would be the following 2 items.
Okay, so those 2 people, did they request this from your agency and this is assigned to you?
Yes.
Okay. And those 2 people never requested anything prior to March of 2021?
From me, no.
Okay. Anybody else in your agency?
I would have to look at the assignments. I know there were several assignments for this particular case.
Okay. Are you the only person who was ever requested to test for bleach?
Yes.
Okay. And you were asked a little bit about, how the elements, sunlight, water may, dilute bleach. You remember that line of questioning?
Yes.
For example, I mean, you would expect a car with its windows up to not be exposed by sunlight or water. Would you agree with that statement?
Potentially water, sunlight through the windows, maybe?
Sure. But the point here is that nobody ever gave you like a cutting or a swab of, fabric from a car, right?
Correct.
I mean, you weren't given any other piece of clothing from Mr. Dinkins himself or anybody else. Just so I'm very clear.
I was only given the items of clothing that were in this report.
Okay, that's all I have. Thank you.
That's what he asked you.
Mr. Stepanski, relative to your findings, you did detect the presence of elements in the fabric that would be consistent with elements in bleach. Would that be fair to say?
Yes, there were elements that could be found in bleach as well.
Okay. And with respect to the pH balance test that you ran, you came up with a pH balance of 7.
Correct.
Bleach would have a pH balance of 10.
Correct. Keeping
How-
in mind that water is 7, so neutral.
Right. And certainly the extraction process would dilute the pH balance that you would have engaged in.
Potentially it could. It's a consideration.
And then even being exposed to environmental factors can impact that pH balance, correct?
Yes.
And then if there was a light blue that was noted with bleach, it would have been a darker blue, correct?
Correct. I do want to specify though, because we're getting into the specifics of my actual testing, that there are other explanations for these color changes and for the pH. For example, the color change could be explained by nitrates, and there was the presence of soil on the items. So I just want to be very clear that just because it changes colors does not indicate specifically that chemical.
Certainly. And then when you talk about the issue of nitrates, with the presence of nitrates, how would that impact the development of any color that you would see in the fabric?
As far as I know, nitrates themselves would not change the color of the fabric.
No, specifically what I meant is when you're performing a color test, how does the presence of nitrates impact results?
Sorry, I misunderstood. It would change the color to a blue.
Okay, all right. So that's also a possibility as well.
Yes.
So we're looking at at least two possibilities with the color test. Would that be fair to say?
Yes.
And of course, this shirt had been recovered outdoors, and there was certainly dirt within the fabric itself.
Yes.
Okay, I have no further questions.
You may sit down.
All right now.
Your Honor, the state calls criminalist Mike Schmidt to the stand.
Now, who is this one? Yeah, that was the tracing analysis. This prosecutor right here, she be on the wall. This lady right here. Woo! She making sure everybody do their job. Like her. I know he running out of there, but them men be running out of there. But let's look at Mr. Henry. Cameraman, you could put it on Henry. Why you didn't put it on Henry? See how he doing on a Friday. These people
Do you swear to tell the truth, the whole truth, nothing but the truth?
Yes, I do.
Please have a seat.
It's a tight one, Mike, but I think you know that. Please state your name for the record and spell both your first and your last name.
Michael Nathan Schmidt, Michael Schschmidt.
Mr. Schmidt, where are you employed?
I work at the Iowa Division of Criminal Investigation Criminalistics Laboratory in Ankeny, Iowa.
What is your title there at the laboratory?
I'm a criminalist,
How long have you been employed at the DCI laboratory?
25 years.
Which section of the laboratory are you assigned to?
I'm assigned to the casework section of the crime lab.
And then do you do analysis on items for the presence of DNA then?
Yes,
Could you describe your educational background?
I attended the University of St. Thomas in St. Paul, Minnesota. I received a Bachelor of Arts degree in biology with minors in chemistry and psychology.
When did you complete your formal education?
1997.
All right, and I'm doing the math. So then would you have started with the DCI after you concluded your formal education?
I had a full-time job as a criminalist in the DNA section of the lab in June of 1998, and I've been there since then.
Thank you. When you started with the DCI, describe the training that you received.
I had about 6 months of training by the senior criminalists in the DNA section of the crime lab. Those were people that had been working there for decades. I was trained in the DNA analysis methods, examining evidence for blood stains and semen stains. And also I was sent to classes. One was at the
You
company
better
that
get ready.
manufactures the DNA kits that we use in the crime lab. They were in San Francisco, California at the time. I had a week-long course in PCR, the polymerase chain reaction, DNA amplification and typing. And I've had continuing education at least 8 hours every year since then, with the exception of 2001 when there were budget cuts.
What is DNA?
DNA is the building block of life. It's found in the nucleus of every cell in your body that has a nucleus. If you arrange it a certain way, the end result is a tree. If you arrange it a different way, you'll have an insect or even a human.
Okay, where do you find, or how do you examine items for the presence of DNA?
In the lab, with some evidence, you can see items that are stained that have blood stem. We look at a lot of blood stains in the crime lab. We also work a lot of sex assaults, and sometimes you can see semen stains with your naked eye, but a lot of times you can't. You have to use an alternate light source or a forensic light to help you visualize the stain.
Come on.
We also examine some items that might have your saliva on them. For example, in burglaries, people leave cigarette butts or pop bottles or beer cans behind.
Come on,
We
lady.
can swab
That's
the
cute.
mouth area of those items and attempt to develop DNA profile from saliva or cells that are left behind on that item.
Okay, now before we talk about the processing of items for the presence of DNA, first of all, educate us about where we get our DNA.
Half of your DNA you get from your mother, and the other half you get from your father, your biological mother and father. It's passed down to you that way. A sperm cell is going to have half of your DNA, and the egg that the mother has the other half of the DNA. They come together, and you have the full 48 set of chromosomes that make up your DNA.
Okay, when you receive items in the laboratory to test for the presence of DNA, then how is it that you, let me do this. Let's talk about the steps that you take then to process DNA samples.
First, I'll examine the actual item of evidence in the lab, basically looking for semen stains or blood stains most of the time. Then I'll take a small cutting of that stain, put that cutting in a small plastic test tube, and add some chemicals to that, heat it up, and that breaks the cells open, releasing the DNA into a solution that we can use. Once I have the DNA into a solution, I'll do what's called quantitation. And quantitation tells us how much DNA is there. The testing we do right now at the lab requires a very specific amount of DNA, a target of approximately 1 nanogram of DNA. So that's why we need to quantitate the samples before we amplify the DNA that is there. The next step in the process is
And then can I ask a question relative to the topic of quantification? So if there is not a sufficient sample, then does the analysis essentially stop there?
Once we start our DNA process, if we do a quantitation, we usually carry it all the way through. Other crime labs in the country, they will stop it if the quantitation is telling them there's not a sufficient amount or there's no DNA there. We are working towards developing that and implementing that, but we are not there yet.
All right. All right. So then after the quantification stage, describe then what's involved in amplification.
Amplification is taking the DNA that's there and making many copies of it. If you imagine a copy machine, starting with you hit the copy button about 1 million times. So that's kind of what we're doing with the amplification process in DNA. We need enough DNA that can be detected by our analysis instrument.
All right. And then once you've gone through the amplification process, then what's the next step before you start the process of comparison?
We load the amplified DNA onto an analysis instrument, and that instrument measures differences in lengths of fragments of DNA. Basically, we're looking at 21 different locations in a person's DNA on their chromosomes, looking for DNA factors there. And the instrument, most of the fragments are between 60 and 460 base pairs long. And there's a laser in the instrument that can measure differences in length as the DNA travels through a capillary or a small glass tube and passes through the detection window where the laser shines on it and takes all the measurements for us.
Now, Mr. Smith, speak to this particular topic. Recognizing that we get half of our DNA from our father and half of our DNA from our mother, I pose the question, is DNA unique to each of us as individuals?
If you look at enough different locations, most people are going to have a different DNA profile. The exception would be identical twins. They're going to have the exact same DNA profile.
Okay, all right. And then once you have developed areas of the DNA to test, then how would you compare DNA of an individual to any locations of items of evidence for the presence of DNA to determine if there is a match?
I will have a printout of the crime scene evidence DNA profile developed from a crime scene, and then I'll have another piece of paper with a profile printed out of potential or alleged victims and suspects in cases, and I will do a side-by-side comparison to see if I can either match or make an elimination between the samples.
That being said, I want to direct your attention to a death investigation. Specifically arising out of a report of a child identified as Breasia Terrell, who was seen in July of 2020, and then we had recovery of remains on March 22nd of 2021. Did Rick Ron of the Iowa DCI reach out to the DCI laboratory to assist in identifying skeletal remains that had been found and then collected on March 23rd of 2021 out north of Clinton, and I know you probably aren't aware of the area, but did he contact us identify who those remains belonged to?
Yes.
All right. Now, did you perform a number of different analyses after the recovery of those skeletal remains?
Yes.
As far as your first involvement in this case, what were you tasked with?
First, the body had been found, and they asked the DCI lab to assist in the identification of the body. So the lab received a bone from the remains, and I was assigned with trying to develop a DNA profile from that bone.
Your Honor, may the record reflect that I'm showing counsel what's been marked as State's Exhibit 57. May I approach, Your Honor?
You may.
I'm handing you what's been marked as State's Exhibit 57 for purposes of identification. I'd like for you to look at that report.
Yes, this was the first DNA report I wrote for this case.
Okay. How is that document captioned at the top in the middle?
Official report of Iowa Department of Public Safety, DCI Criminalistics Laboratory.
Is the lab case number attached to your report that was assigned to this investigation and submissions that came in to the DCI for analysis?
Yes, the lab case number was 2020-16859.
And as far as varying reports that were generated by the DCI, what report number is assigned to this report?
report 1.
What date did you complete your findings and author this report?
March 30th, 2021.
And does your report identify what type of report this is?
Yes, this is a DNA report.
And is your signature attached to this document as the criminalist who did the DNA analysis?
Yes.
Your Honor, the state would move to introduce State's Exhibit 57.
Exhibit 57 will be admitted.
May I publish, Your Honor?
You may.
All right. In looking at State's Exhibit 57, what we're doing, Mr. Schmidt, is just trying to give everybody a perspective of what came in so that we know what items of evidence that you were working with. Do you have 3 separate sections showing 3 different submissions that came in?
Yes.
All right. The first entry, give us the information as to date and what individual with what agency had submitted items and what those items consisted of.
Okay, on March 25th of 2021, Mark Bethel with the State Medical Examiner's Office submitted a shirt, shorts, a bra, and a box containing a left femur bone.
All right. For our next section, what was our submission date for the second item of evidence and who was responsible for that submission?
On March 25th of 2021, Mark Bethel from the State Medical Examiner's Office submitted an item containing tissue from the left leg.
All right. And are we talking about the earlier submission of the box of left femur? Would that tissue have come from that?
The tissue came from the left leg.
Gotcha.
It was in a small plastic test tube.
Okay. And then for our third submission, what is the date? Who made that submission and what item was that?
On March 26, 2021, Evan Obert with the Davenport Police Department submitted the known buccal swab from Aisha Langford.
All right.
From
Let's
the
talk
mom.
about the known buccal swab of Aisha Langford. We're going to go back in our education so that we can set our perspective. I would represent to you that Aisha Langford is the mother of Breasia Terrell.
Mhm.
Now, what is a buccal swab?
Buccal swab is basically a cotton-tipped swab, basically a Q-tip, except it just has cotton at one end of the swab, not both. Now, to get a known DNA sample from someone, an officer, a detective, or a sheriff's deputy, we'll take that swab and rub it on the inside of a person's mouth against their cheek or their cheek wall. There are a lot of cells present in that. It's usually plenty of DNA for us that we can develop a DNA profile from that swab.
When the swab from Aisha Langford came into the DCI laboratory, did you in fact develop a DNA profile for Miss Langford?
Yes, I did.
All right. Now, why did you need to develop a DNA profile from Miss Langford?
In order to make a comparison to the unidentified remains that were found in this case, we do have a DNA database that we have access to. It's a national DNA database, but it contains DNA from crime scenes and convicted offenders. I could, if we didn't know who the person's mom was, we had no idea who the body was, I could put that profile from the body into the database and see if there was a match somewhere. But most of the time, we do ask for known samples from the decedent's possible parents or their children in order to make a comparison. We basically don't want an innocent person's DNA in the database if we don't have to, if we can make an identification in another way. And basically, children aren't going to be in our DNA database.
Certainly. And then did you take the tissue from the left leg and develop a DNA strand from that?
I attempted to, that I was unable to develop a DNA profile from the tissue sample from the left
Okay.
leg.
And then can you explain why?
When a body is left out in the Iowa and seasons pass, that body is subjected to the extreme weather, sun, rain, snow, ice, wind, the heat. If if you ever find a dead bird in your yard when you're mowing the lawn, a couple days later, it stinks really bad and it decomposes really fast. That's just the way Iowa weather is. Now, in other places, it's not like that. Like in Arizona in the desert, the bodies dry out pretty quickly and are kind of preserved a lot better than they are here in the Midwest. And so when bodies are subjected to those extreme conditions, they decompose, and then therefore the cells also decompose, and the DNA in those cells degrades and eventually gets to the point where we can't detect the DNA that's there because it's been either completely degraded, inhibited, or destroyed.
Okay, so then does that take us to the left femur? Were you able to develop any DNA from the left femur?
Yes, I was.
All right, and describe.
I took a cutting from a section of the femur from the adolescent doe, and was able to develop a DNA profile from that. Based on the comparison of the DNA profiles of adolescent doe and Aisha Langford, adolescent doe could not be eliminated as a possible daughter of Aisha Langford. The probability of kinship is 99.9998% as compared to randomly chosen, unrelated, untested individuals.
Okay. Now speak to the language that you've used in your report where you indicate that she could not be eliminated as a possible daughter. And this is just for sake of discussion. Let's assume for sake of discussion that Aisha Langford had 3 daughters. When you report this type of finding, what is the import of this type of finding if she were to have 3 daughters?
I would need the other 2 daughters to determine if she could be included as a possible mother of those 2 daughters. I can say that Aisha Langford is definitely included as a possible mother of the unidentified remains in this case.
Okay. Were those findings then reported to the state medical examiner's office?
Yes.
And then so with that, since Aisha Langford was identified as a biological mother of this female child, would that then require the state medical examiner's office to obtain dental records to make a definitive determination as to which these skeletal remains were?
Yes. My role is to develop DNA profiles and do the comparisons and issue my report. In body identification cases, the medical examiner or state medical examiner has jurisdiction over the body. They are the ones that ultimately make the identification decision. Even when we have a match in a database, we still issue our DNA report to the medical examiner, and they decide whether the body has been identified or not.
Okay, let me go ahead and take that exhibit from you. We'll go on to the discussion of our next report. How many reports in total did you author?
3 for this case, 2 others on a cross-referenced case.
Okay, and we'll set the discussion aside for the cross-reference case a little bit later on. We've had discussions about that. But as far as your second report, Your Honor, may the record reflect that I'm showing counsel what's been marked as State's Exhibit number 58. I'm handing you what's been marked as State's Exhibit number 58 for purposes of identification. Do you recognize State's Exhibit number 58?
Yes.
How do you recognize State's Exhibit 58?
This was a DNA report that I issued on April 19th of 2021.
And in this report, what is it that you will be speaking to?
This was the shirt, shorts, and bra submitted by Mark Bethel from the medical examiner's office.
All right. Your Honor, well, let me ask you this. What report number is this?
5.
Does it bear the same lab case number?
Yes.
What date did you generate this report?
April 19th of 2021.
And again, does it identify the nature of the report that you authored?
Yes, it's a DNA report.
And does your signature appear at the back of the document as the criminalist who did this analysis and generated this report?
Yes.
Your Honor, the state would move to introduce State's Exhibit 58.
No objection.
Exhibit 58 will be admitted.
May I publish, Your Honor?
You may.
All right. So as we look at State's Exhibit 58, then are we simply focusing on the 3 items of evidence for that first submission that we saw in the earlier report,
Yes.
okay? And for purposes of our written record, just establish that again.
This was a shirt, shorts, and bra submitted by Mark Bethel from the medical examiner's office on March 25th, 2021.
Okay. Now describe the type of DNA analysis that you were doing on these articles of clothing.
These articles of clothing, I was screening for evidence of sperm or seminal fluid, basically sex abuse type evidence.
Okay. Mr. Schmidt, can you speak to the impact of environmental factors if in fact there was sperm or seminal fluid on an article of clothing that had been located outdoors over the course of multiple seasons?
The chances of me being able to develop a DNA profile from that type of item, if it's left outside in Iowa weather for multiple seasons, are pretty slim for me developing any kind of DNA profile from it. The reason I was able to identify or develop a profile from the bone for this case is because the DNA is basically a lot more stable in bones in bodies. The DNA is trapped in the crystallized calcium structure of the bone, and it takes a lot of time, a lot of energy to release the DNA out of those crystallized, hardened, calcified bone cells.
All right. Can you also speak to the topic of bleach? How would bleach impact the ability to detect sperm or seminal fluid on an article of clothing if it was poured over that clothing?
Bleach, it destroys DNA. We use a solution, 10% bleach in our lab to clean our work areas. And that's just a 10% diluted bleach solution, but that's good enough to clean our working surfaces, to clean the surfaces of any DNA. That way we're not contaminating items in between examinations.
Okay, all right, so walk us through your findings in this particular case.
I attempted testing on 2 stained areas of the shirt. Both screening tests were negative for seminal fluid in those areas, and I did not find any sperm microscopically in either of those samples. I attempted DNA analysis on the sperm and epithelial fractions for both cuttings. I was unable to develop any DNA results from the shirt.
I want to stop you right there. Now, you have mentioned 2 terms. You talk about the sperm fraction. It's obvious, but define.
When we look at a sample that contains seminal fluid or possibly contains seminal fluid, we attempt to separate out 2 different fractions from that sample. In the end result, we're going to have, or attempt to have, 1 tube that has DNA from skin cells or blood or saliva. And then in another tube, we're going to have just the sperm cells or DNA from sperm cells that are there. Our lab calls those the sperm fraction and the epithelial fraction. Other forensic labs call those the forensic 1 fraction or F1 and the forensic 2 fraction or F2. It's just a naming thing. Just because I call this sperm fraction doesn't mean it contains sperm there. And that's probably why the other labs call it an F1 fraction, because it doesn't always contain sperm.
Okay, and so then that explains the difference between sperm fraction and the epithelial fraction.
Yes.
All right. Now, let's also speak to this term, spermatozoa. Tell us what you mean by spermatozoa.
Spermatozoa is just a plural for sperm cells. If there's just 1 sperm cell, then we'll call that a spermatozoon. If there's 2 or more, then we will say spermatozoa.
Okay, then let's go back and let's circle the wagons and start where we were at before I had you give us those definitions of a sperm fraction versus an epithelial fraction.
Okay. I was unable to develop any DNA profiles from either the sperm or epithelial fractions of both samples from the shirt. I also tested a stained area of the crotch liner of the shorts, and a stained area from the seat area on the interior liner of the shorts. Again, for those two samples, both of those samples screened negative for the presence of seminal fluid. I did not find any sperm in either of those samples, and I was unable to develop DNA profiles from the sperm or epithelial fractions of both samples.
I want to make sure that we understand what you're saying. All right, when you say that you were unable to identify any sperm as being present, again, if clothing is outdoors and is subjected to the elements, will that destroy the sperm?
It can.
All right, so then when you say that you were unable to identify sperm, do we have to consider the environment within which those articles of clothing were located and what they had been subjected or exposed to?
Yes.
Okay, continue.
I also examined a bra that was found in two pieces. I attempted DNA analysis on the interior cups of the bra. I also screened the cuttings that I took from the cups of the bra. Both of those screened negative for seminal fluid, and no sperm were found in either sample. And then again, I was unable to develop any DNA profiles from the sperm or epithelial fractions of both of the cup samples.
Okay. Are these findings consistent with what you would expect to find if you had articles of clothing laying outdoors through a summer, fall, winter, and spring month?
Yes.
Okay. Let's talk about your third report that you generated. Your Honor, may the record reflect that I'm showing counsel what's been marked as State's Exhibit 59.
Wow.
I'm handing you State's Exhibit 59. Do you recognize State's Exhibit 59?
Yes.
How do you recognize State's Exhibit 59?
This was the DNA report that I issued on July 29th of 2021.
Okay. And again, does this contain the same lab case number that you've previously referenced?
Yes, 2020-16859.
What report number is this?
7,
What date did you generate the report?
July 29th of 2021.
What type of report is this?
This is a DNA report.
And does your name appear on this document as the author?
Yes.
Your Honor, at this time, the state would move to introduce State's Exhibit number 59.
No objection.
Exhibit 59 will be admitted.
Your Honor, again, may I publish?
You may.
It's interesting, y'all. Got to pay attention to the words he's saying.
What was the nature of the submission as noted in State's Exhibit 59?
This was an additional submission from Mark Bethel with the State Medical Examiner's Office on June 30th, 2021. Mark submitted fingernails, or apparent fingernails.
Okay. Now, what type of fingernails did you say that he submitted? Did you say parent fingernails? I'm not sure that I understood you correctly.
Yes, I couldn't determine if they were real fingernails or the fake fingernails.
Oh, I understand what you're saying.
Oh, okay.
What was the nature of the request then that was made for the fingernails that had come from the State Medical Examiner's Office?
Just if I could develop any DNA profiles from the fingernails.
Okay, and then describe what you were able to determine or not.
Up a DNA profile from the swabs of the fingernails.
Okay, thank you.
Come on, we need some evidence.
Take that report from you.
Henry?
I'd like to set the stage for our further discussion. Now, were you asked to do DNA analysis on some items of evidence, which would have included a white medium tank top as a result of a murder that DCI agent Rick Ron was involved with? It was a collateral well, I shouldn't say collateral, a completely separate investigation from the investigation of Breasia Terrell.
Yes, there was a separate homicide from Clinton County that the DCI was investigating.
Okay, and I think what I'll do, what's her last name? Your Honor, may the record reflect that I'm showing counsel what has been marked as State's Exhibit 157.
This interesting, y'all. Pay attention. Did it answer any question you asked? Thus far, leaving the clothing out
May I
there.
approach, Your Honor?
You may.
I'm handing you State's Exhibit number 157 for purposes of identification. Do you recognize State's Exhibit 157?
Yes,
How do you recognize State's Exhibit 157?
this is a DNA report that I wrote January 21st of 2023.
All right. What is the case number associated for this laboratory case number?
The DCI lab case number for this case is 2020-17232.
Is that a completely different lab case number as compared to the lab case number assigned to the Breasia-Terrell case?
Yes, it is.
All right. So then let's just go back. For the Breasia-Terrell's case, what was our lab case number again?
2020-16859.
All right. Now, by virtue of that homicide that had occurred in Clinton, was the lab doing a number of different analysis based on submissions that came in through that case?
Yes,
Okay. Was some of the evidence collected in that investigation turned over to Davenport just to submit to see if there was any connection at all with this case?
yes.
All right. And then as we look at this, what date was this report generated again?
January 21st of 2023,
What type of report?
a DNA report.
Does your name appear as the criminalist on this report?
Yes,
And set forth the listing of items that came in through Davenport asking you to test.
yes.
Your Honor, the state would move to introduce State's Exhibit number 157.
No objection.
Exhibit 157 will be admitted.
Okay. May I publish, Your Honor?
You may.
All right. Now, I'm putting forth State's Exhibit 157 under the Elmo. If you could, let's again go back to the section of the report that gives us a date, who the submitting officer was, and a description of the items that came in.
On November 28th, 2022, A. Poirier with Davenport Police Department submitted a Hanes crew-style white medium t-shirt, one children's white medium tank top, and some hairs sealed in tape.
All right. If the body in this particular case was recovered on March 22nd or discovered on March 22nd of 2021, then this report was authored when?
January 21st of 2023.
All right. And do you know when that particular homicide took place?
I do not.
That's okay. When these items came into the laboratory, what were you asked to do to see if it had any tire connection to the Breasia-Terrell investigation?
There were some small shirts, child-sized shirts. So I was just asked to see if there was any DNA that could be either eliminated or matched to Breasia-Terrell and Henry Dinkins.
Was there some type of blood or other bodily fluids on that child's t-shirt?
Yes.
What was it?
There was blood on the child t-shirt in 3 stained areas, DCI item 2. I was able to develop DNA profiles from those areas. The DNA profile developed came from a male source. And later on, Henry Dinkins was eliminated as the source of the DNA profile, and Breasia-Terrell was also eliminated as the source of the DNA profile.
And so the male source, how is he designated in this report?
Male A.
And has male A been identified to date as who the contributor would be
No,
for that blood on that child's t-shirt?
no, he wasn't.
Well,
Does that conclude your involvement in this matter?
this is
Yes.
crazy.
I have no further questions, Your Honor.
That's why I asked y'all
Mr.
who
Waters.
else.
Mr. Schmidt, you were asked a line of questions about how the elements, sunlight, rain, snow, whatever it may be, is or has the ability to destroy DNA. Do you remember that line of questioning?
Yes,
Okay. So you would agree then that those elements can destroy DNA, sperm, whatever it may be?
yes,
Okay, isn't it just as explainable that there was just never sperm or DNA on those shirts?
yes, I did not find sperm on those shirts, so that's a possibility, yes.
Wouldn't that just be the easier explanation?
It, you could go either way on that.
Sure.
There's no way for me to know that. I can only say that I didn't find anything
Yeah,
there.
and that's fair. You know, I'm not trying to trip you up, but then what we just talked about, those shirts, I think it was found from a black Challenger, a car, right? Do you remember that?
It was found in a car. I know
Okay.
that.
And I think it was just maybe one shirt or two shirts.
Two shirts.
And was blood visible on those shirts?
Stains were visible to me. It didn't look like blood, but it did screen positive for blood. Could have been vomit. We don't have a screening test for vomit. It could have been diarrhea. There's just, all I can say is the stain screened positive. The stains screened positive for blood.
Sure. And then your assignment with those items that we just looked at was simply to test or compare the DNA from male A to Henry Dinkins and Breasia Terrell. Am I understanding that correctly?
That's right.
Okay. At no other time did somebody request to look at any other person of interest other than Breasia Terrell and Henry Dinkins?
No. Had I developed an unidentified male profile from Breage's clothing, especially if it came from a sperm fraction or sperm DNA, and Henry Dinkins was eliminated, I would have put that profile into CODIS because there shouldn't be sperm on a child's clothing. That would at least be an act of sex abuse. And so that profile would have been put into the database and would have eventually been compared to suspects throughout the nation. So it's just that in this case, I was only given knowns from two people.
Sure. And male A, we don't know who that is.
That's right.
Okay. That's all I have.
Just kidding, yeah.
Yes, thank you. Mr. Schmidt, do we also have to recognize that you didn't find Breage's DNA on any of the articles of clothing?
That's right.
Okay, and I'm talking about the articles of clothing to include the shirt, shorts, and bra that were found out in the elements on March 23rd of 2021.
Yes, I didn't find her DNA on any of the clothes from either of the cases.
Okay, and so under that circumstance, again, presumably if an individual's wearing clothing, is there going to be a transfer of our DNA on that clothing?
Usually, yes.
All right, and with those articles being out in the elements, does that help us to understand why you would not be able to identify any of Breage's DNA on those articles of clothing?
Yes.
I have no further questions.
You may sit down.
Thank you.
That was good. Yeah, that was okay. I'm gonna stop it right
Your
here.
Honor, this is all of the evidence that the state has to offer at this time. As we indicated yesterday, Detective Hammis would be our last witness, but she's going to take a significant period of time, we think.
I am going.
Thank
to
you very
stop
much.
Right here.
I wouldn't, is the defense asking that we start at 9:30 again on Monday?
What's going on?
We will.
Thank you. Thank you.
You're welcome.
All right.
Have a nice weekend, everyone.
Thank you.
String God, come on now. I'm trying to end my presentation.